Hi, in this lecture, we're going to go into some muscle physiology and we're going to look at how muscle contraction occurs. I highly recommend that you watch some of the helpful links that I have posted in Blackboard and that you go into Mastering A&P and go into the study area. I showed you some of these videos previously, but there's some excellent big picture animations and interactive physiology that will take you through this process step by step. It can be a little tricky. So first of all, just electrophysiology is it's a study of electrical charges and electrical changes. We have cell membranes, and muscle and neurons are very excitable. And what that means is that there's a charge across the cell membrane, and you'll see in this picture that the inside of this cell is actually negative and the outside of it is positive. So we have a separation of charges. We have an electrical condition. And when things change, when we allow maybe positive ions to come in, if we flip the charge from the inside and make it positive, now we're changing that charge and it kind of shoots all the way through a cell, kind of like electricity shooting down a wire. And these changes where we make the inside positive or we make the inside negative are all electrical changes, which is how cells communicate. So there's something called an RMP, which stands for resting membrane potential. It's called potential because it's actually a form of potential energy. So your electrical cells have potential energy at rest, and it's due to the charge across the membrane. So as I mentioned, if we were to measure with a little voltmeter the charge inside the cell, remember the cytosol or the ICF, that charge is always negative. Now the actual number might vary. Muscle might be minus 90, a neuron might be minus 70. The number doesn't matter right now. What matters is it's always negative, and the outside of the cell has a positive charge. So we have a separation of charges. So we say the cell is polarized. So polarized means there's a separation of charges across the membrane. Well, we kind of want to know, why is the inside negative? What's going on with that cell? Well, there's a couple things that happen here. First of all, you have an ion concentration gradient. You have a difference in your potassium and sodium. These are the main cations. So remember, cation means it has a positive charge, and you have a lot more potassium inside the cell, and then you're going to notice that you have a lot more sodium on the outside of the cell. So we have these different concentrations that differ, whether they're in the cell or out of the cell. Another thing we have with cells is that inside the cell, you'll see here, there's also a lot of proteins. And proteins have a negative charge. So those are important. So we do have a bunch of negatively charged molecules within the cell. Now we focus on those cations. We focus on sodium and potassium, and I'll show you why it's the cations movement that makes the charge negative inside the cell. In Bio 119, this might look familiar. This was out of your textbook. We have two types of channels. A channel is like an opening. You can see here, right? A little protein opening, little channel across the cell membrane, because cell membranes are selectively permeable. And what we have are leak channels. Now, leak channels are called leak channels because they are always open. So they're leaky. Notice they're always open. Now, they're very specific. You'll have a leak channel for potassium, and then you will have leak channels that are specific to sodium, and they're always open. And so what happens is these ions, these cations, are going to always follow their concentration gradient and use diffusion. So remember that there's always a lot more sodium outside of the cell. So if I have a little leak channel, an opening, sodium's always going to want to come inside that cell. Now, if I look at potassium, I have potassium leak channels, but remember there's a lot more potassium inside the cell. So if there's a little open door, a little channel, potassium's always going to want to leave. So the ions always follow their concentration gradient. They have to. They're going to move from where there's a higher concentration to a lower, and the leak channels are always leaky. Now, if you look at this picture, you might notice that there's a lot more leak channels for potassium than there are for sodium. And so we say that the cell membrane is more permeable to potassium than it is to sodium because there's more leak channels. So more potassium can leak out than sodium can leak back in. Now, gated channels are different. Gated channels are only going to open or close in response to a specific stimulus. So some things that could open a gated channel might be a chemical, like a neurotransmitter could bind and open or close these channels, or the voltage. So we have a charge across the cell membrane, and some of these channels open or close depending on what that number is, depending on what that charge is. So I mentioned we have these electrochemical gradients, and what that means, it's a fancy way of saying that if we are to look at potassium, so I will switch to orange like the picture. I have all my potassiums inside the cell. Anytime I have a leak channel, right, potassium always wants to leave. It has a gradient. So it has a gradient, a diffusion gradient, or a concentration gradient where it wants to leave the cell. So if I have a container and I have a bunch of positive charges in it, I'll just put 5, and then let's just put a couple negatives, because we do have some negative ions inside the cell. So let's say in this example, notice I have 5 positives and 2 negatives, right? Well, let's say that now 4 of those positive charges leave. So let's get rid of 4 of them. So I'll get rid of that one. Okay, now notice what happened to the charge inside my bucket, right? In my example, see how now it's more negative? I didn't move the negatives. I didn't move those proteins. What I did, though, was I removed positively charged ions. And as I remove the positives, then that's going to make the inside more negative. And that's where I'm headed with this. So we know that we're much more permeable to potassium, and potassium's going to want to sneak out. Now, diffusion happens until equilibrium is reached. So if we let that happen until equilibrium was reached, the charge would actually be about minus 90 millivolts across the cell, which is pretty negative for a muscle cell. So roughly around minus 90-ish. Now, and you don't have to memorize that number. Now, if we are looking at the sodium, we said that sodium, it has a lot more sodium outside of the cell. So sodium has an electrical chemical gradient. It has a chemical gradient because it has a concentration gradient, because there's more sodium on the outside and less on the inside. It also has a chemical gradient because sodium has a positive charge and the inside of that cell has a negative charge. So what I want you to understand is that sodium really, really wants to get inside that cell. And if sodium starts getting inside that cell, now we start adding positive charges within. So in my example back here, if I start putting sodium back in, do you see how that will ultimately change my charge and make it more positive? So we're really looking at where the positive charges go, and if more leave than come in, then you're going to have a net negative charge on the inside. And that's exactly what's going to happen here. So because we have more of these leak channels for potassium, and we said earlier the cell membrane is more permeable to potassium, that means that more potassium is going to leave that cell than sodium can get back in. So more potassium is going to leave, very little sodium gets back in, and that's going to create the negative charge on the inside of the cell. So you're losing those positive ions like I showed you previously. So again, just to do this really quick, let's say we have a solution, and let's just make 4 negatives, and I will use purple for, let's use purple now for potassium. So these are going to be my positives, are going to be my potassium. Okay, so I've got 1, 2, 3, 4, 5. Looks like I have 6 potassium there. So now, and let's just go ahead and draw the sodium while I got my pen handy. I'll do the sodium out here. So I'll do my little Na, and I'm just going to use the pluses here. So we have a lot of sodium out here. So let's say, and again, this isn't scientifically precise, I'm just trying to give you an example. So let's say that we're going to have some of these potassiums leave. So let's say that 4 potassiums leave. So I'm going to knock out 4, 2, 3, 4. Okay. Notice my net charge in my cell right now. I have 4 negatives, 2 positives. So my net charge is minus 2, right? Okay, but now we're going to let a little bit of sodium in. So let's just say 1 sodium comes in. Well, that's going to, again, my charge is still negative. right? But now it might be -1. So what happens is, because I'm losing so many potassiums, and it's on a much larger magnitude, oops, sorry about that. Because I'm losing so many of these potassiums, they're leaving, I'm losing more positive charges than I'm replacing with sodium. And that's going to cause a net negative charge across that cell membrane. So the inside will be negative. Now, we don't want to just let this happen forever, because remember, if potassium keeps leaving, it could make it too negative. And if sodium comes in, it could make it positive. And some of our cells, we might keep it like -85 millivolts or -70 millivolts. So they all have their own resting membrane potential. So the sodium potassium pump's job is to operate at rest to basically kick any of that sodium that got in out and pull that potassium back in. So this is active transport. It's moving the ions against the concentration gradient, and it uses a lot of ATP. In fact, if you've ever heard of your basal metabolic rate, just the amount of energy for these pumps to run, right? If you're to lay in bed all day, it'd be about 1,200 calories, 1,000 to 1,200 calories. Most of that is to operate these pumps, so we can keep our resting membrane potential where it's supposed to be. So it's negative because of the cell membrane permeability. It's more permeable to potassium than sodium. So more positive charges leave, and more potassium leaves than is replaced by sodium. The reason why it's maintained is because of the sodium-potassium pump. And it's very, very important that we keep that RMP just right. So then what is an action potential? An action potential is when we flip the charge. This is how cells communicate. This is electrical communication. And this is going to be very important in muscle and neurons. Now, when we're reversing the charges, this time we're going to use the gated channels. And remember, the gated channels are only going to open or close in response to something specific, like maybe a neurotransmitter or the charge across the membrane. So we'll be looking at some voltage gated channels as we go over what an action potential is. So very basically, if you have your RMP, in this example, they're using -90 as the RMP. So that's just where you're kind of hanging out. Something happens. So you have a stimulus, and we usually call this a threshold stimulus. And when you get a threshold stimulus, what that's going to do is it's going to open up the sodium gated channels. So in this picture, see the little sodium gated channel? It's closed. Nothing's getting through. But when there's a stimulus, it opens up that gated channel, and now sodium follows its concentration gradient. It comes rushing inside that cell. As sodium enters the cell, the charge starts to become more positive. And this is called depolarization, because it's not polar anymore. It's changing it. It's becoming positive. So it's the voltage-gated sodium channels that open. And when they open, sodium rushes into the cell. So we always say threshold has to occur. And all that means is if you have a threshold stimulus, it's going to be strong enough to open those gated channels. Now we've depolarized. What happens though is that once we reach +30, that's the stimulus to close those voltage gated sodium channels. So it's a little messy here. They close the voltage gated sodium channels. So remember, they're going to open and close at a certain charge. So what's going to happen for number 2 on your paper is the voltage gated sodium channels will close. And again, just once enough sodium rushes in to change that charge, now they're going to close. Well, when they're closed, no more sodium gets in, so we don't go beyond that number. The third thing that's going to happen is the repolarization. So now we need to reset it. And now for repolarization, the voltage gated, so we're voltage gated potassium channels open. So at +30 millivolts, the sodium channels close, the potassium gated channels open. And whenever a potassium channel is open, potassium leaves the cell. Remember, it follows its diffusion gradient. Now we're losing charges and we reset. We return back to our RMP so we can do it one more time again. And so this action potential, this changing of sodium rushing in and then potassium rushing out as it travels through the membrane is called an action potential or a nerve impulse. So if I go back to our original picture, what they're trying to show you here is that it just kind of comes in and that just passes all the way through. The sodium comes in and the potassium leaves, and it's like a wave of electricity or current that's going to travel all the way across the cell membrane. So we have something called the all or none law, and all that means is you have to have a threshold stimulus. Remember, the threshold stimulus is what's going to open those sodium voltage gated channels. So if it's threshold, they all open, you depolarize, the gates close, the sodium gates close, the potassium open, and you reset. If it's sub-threshold, nothing happens. So it's called the all or none law. You either get the whole thing or nothing. So a lot of times the example is used pulling the trigger on a gun. It doesn't matter how hard you pull the trigger, the bullet leaves at the same velocity. Well, once you open these sodium channels, then sodium rushes in and you start this whole process. So this electrical information is going to be transmitted from a nerve. Remember that the nerves are going to contain neurons, and the type of neurons we're looking at are called motor neurons because they're carrying information away from the central nervous system. And where the neuron meets the muscle is called the neuromuscular junction. So you can see that skeletal muscle is innervated, it has a nerve supply, and there's something called a motor unit. And a motor unit is one motor neuron and all of the muscle fibers it innervates. Innervate means it supplies, it stimulates. So remember that muscle fibers are muscle cells. So you can have motor units that are pretty big with one motor neuron to like 300 fibers. Now, when we look at a neuron, we looked at this a little bit. Remember that the neuron kind of comes down. You have the axon, and then the axon ends in these like fine branches, and those they were called telodendria. You don't need to worry about that now. But at the end of the axon, how it splits, there were these round things called the axon terminal, or you can call it a synaptic knob. I don't care which one you call it. A lot of times they call it axon terminal. And so inside that's this big yellow thing right here, you're going to have the neurotransmitters. Now, you're going to have these things called a vesicle. So you see the little round vesicle, little membrane enclosed space. And inside the vesicle are the neurotransmitters. And the neurotransmitter we're looking at for muscle is acetylcholine. And we usually just abbreviate it ACH. Now, you'll notice here that where you have the axon terminal or synaptic knob, and then this is the muscle here, that there's always a space. And the space is called the synaptic cleft, or you can call it the synaptic gap. And so that's just the space between the axon terminal and the muscle. We're looking at the actual sarcolemma. And the muscle is this like invaginated area, right? It's got this like curved area. This area on the muscle fiber that has the receptors for acetylcholine is called the motor end plate. So these right here are receptors for acetylcholine, the neurotransmitter. Remember that we're looking at chemicals, so they're going to bind to something specific. They're very, very specific, like a lock and key. So acetylcholine will bind to its receptor, and the area of the sarcolemma that contains the receptors for acetylcholine is called the motor end plate. So here's another view of a motor unit. In this example, we have two. We have one here and one here. So there's going to be numerous motor units. But remember, it's the one motor neuron, and then the neuron branches, and it innervates several muscle fibers. And again, if we look at a close-up, we have the synaptic knob or the axon terminal. Inside are the vesicles with the neurotransmitter, and then you always have that little gap. And here's a close-up of the motor end plate, and you can really see these receptors in here, and those are the receptors for acetylcholine. So, because you have this space, it doesn't directly touch it. So when that neurotransmitter is released, it's going to have to use diffusion, and it's going to have to diffuse across that little gap until it can get to the receptors and bind to them. So let's go over the steps for muscle contraction. So we're going to look at skeletal muscle contraction. Before we go through it, we have to first mention what happens, what's it look like when it's relaxed. So just a few things. When skeletal muscle is relaxed, calcium concentration is high and it's in the SR. Remember, the sarcoplasmic reticulum stores calcium, and the terminal cisterna was like the bulging area right next to the T tubule. That's where most of it is released from. So we got lots of calcium inside there just waiting. We have ATP, and the ATP concentration is high, and it's actually hanging out on the myosin clubhead. So the myosin clubhead has some ATP on it, ready to go. And those myosin clubheads, right, they can't form a cross bridge. They cannot grab actin because the TT complex is in place, right? The cellular chastity belt is blocking those active sites, so it cannot grab it. A lot of times when people teach this, they'll first start talking about this thing called excitation-contraction coupling. It's just a fancy way of talking about how the action potential or nerve impulse comes from the nerve and goes into the muscle. So very basically, you have this wave of electricity shooting down. It's called an action potential or a nerve impulse. It's coming through a nerve. It's coming down the neuron. As it runs down the neuron, it's going to travel through the motor neuron. Remember, that's the type of neuron we're looking at. And it comes all the way down to, again, you call it the axon terminal or the synaptic knob at the neuromuscular junction. Now, when it gets there, something happens with calcium. Calcium ions are actually out in the ECF, extracellular fluid. They enter the axon terminal. When they enter the axon terminal, that causes those vesicles, those synaptic vesicles, to come over and bind to the membrane, to the end of the synaptic knob. And when they do that, that's going to trigger the release of acetylcholine. So it's the calcium ions that enter that causes the synaptic vesicles to release acetylcholine. ACH diffuses across the gap. So you can see the little ACH here. So it diffuses across the synaptic cleft and it binds to the receptors at the motor end plate. So here's the receptors. So if you don't have calcium, you don't release acetylcholine. Important point, right? You need it to trigger the release of acetylcholine. Now, acetylcholine diffuses, it binds to its receptors. When it binds to its receptors, it will then trigger their voltage-gated channels, the opening of these sodium voltage-gated channels. And what you saw previously will happen here. When you open the voltage-gated channels, sodium is going to rush into that cell membrane and flip the charge and depolarize it. So to go through this again with your notes, we said when that action potential comes down, right, comes from your brain, goes through the motor neuron to the axon terminal, and then the neuromuscular junction. We know calcium enters, and when calcium enters, that's what causes that vesicle to fuse, and basically that's what triggers acetylcholine to be released. So calcium enters, ACH exocytosis just means it's released. Acetylcholine has to diffuse across that tiny gap. There's a little bit of time delay here. When it binds to the receptors, right, the chemical binds to the voltage-gated receptors, it opens voltage-gated sodium channels. And then once you open those voltage-gated sodium channels, now sodium rushes in. And just as we saw, you get this wave of electricity, you get this action potential traveling all over the surface of the muscle fiber. It's going right through the sarcolemma and it's heading right to the T tubules. Remember, the T tubules are just those channels, and their job is to guide that action potential all the way down to the SR. So I'm going to play you a little video. It's a little grainy, but I think it's pretty helpful for showing this process to you.
Skeletal muscle, the central nervous system initiates an action potential that travels down the spinal cord to the motor neurons. As the nerve fiber branches, the action potential travels down each branch. Each nerve fiber branches many times and stimulates several skeletal muscle fibers. The union of the axon and muscle fiber is called the neuromuscular junction. Zooming into the microscopic level, each branch of the neuron has a terminal that invaginates the muscle fiber, while remaining outside the muscle fiber plasma membrane. The action potential arrives at the axon terminal. In the terminal, the action potential causes the release of acetylcholine from the synaptic vesicles into space between the axon terminal and the muscle fiber, called the synaptic cleft. In the synaptic cleft, the acetylcholine binds with a receptor site on the fiber membrane, which opens a chemically gated ion channel. Sodium then rushes through the ion channel into the muscle fiber, causing an action potential to form on the fiber membrane. The action potential spreads along the muscle fiber. As more nerve branches activate additional fibers, the action potential spreads over the entire muscle.
So hopefully that was helpful. I know it was a little hard to see, but it was hard to find a good one that I liked. So now we have this action potential is going across the entire sarcolemma. And as it passes through it, it depolarizes it. So now we're in the muscle. So excitation, contraction, this wave, this action potential is in the muscle. It goes down the T tubules. So number 4 in your notes is propagated down the T tubules, they're the channels, all the way to the SR. Remember when we looked at the SR, the sarcoplasmic reticulum, you had that terminal cisternae right next to it, and that's where the calcium ions are stored. It's that action potential that triggers the release of calcium. So you have to excite the muscle, you have to depolarize the muscle, and then that depolarization goes across the cell membrane, down the T tubules, and that's what triggers the release of calcium. Now remember, calcium is the key to the cellular chastity belt. So calcium is going to be released, it's going to leave the SR, and it combines with troponin. So it binds to troponin, and when it does that, it changes the TT complex. It unlocks it, it breaks it down, and it moves out of the way. Now those active sites that myosin wants to get a hold of are there and ready for myosin to grab. So once we've removed the TT complex, notice again, you can see the little yellowish and the blue for your TT complex and the myosin head. It's the calcium that unlocks it. Now those active sites are exposed and myosin can grab actin and form a cross bridge. And here's another view when they're blocked with the TT complex. Now they've been moved and now myosin can grab actin. Now we contract. So we have to have the ATP. We already said that it's there. We know it's there. We know that ATP gets broken down into ADP and a phosphate and energy. And that's the energy that's going to be used a couple times through this, what's called a cross-bridge cycling. The first time we use ATP, so you have ATP hydrolysis. This is ATP hydrolysis. Breaking down ATP to release energy. The first time that happens, it actually helps take this myosin clubhead and it cocks it. It basically allows it to swivel and attach to actin. I always think of this as those Nerf guns. You know how you got to cock them, you got to pull the spring back so that it's ready to shoot those little foam bullets at everybody or foam darts. So it's cocking it. So that's the ATP. Now what happens is once it cocks the head, it basically bends it. Remember, this thing can pivot. Once it pivots and it's attached to myosin, then what happens is it's just going to swivel and do what's called the power stroke. So the phosphate jumps off. And when the phosphate jumps off, it causes that head to kind of relax. But when it relaxes, it actually swivels back. And when it swivels back, it takes actin with it. So it pulls it. The second time we need ATP is we need it to break the attachment. We need it to let go of actin. And so what happens is you have this cocking and grabbing. You have the power stroke where it swivels or pulls, and then you have the release. And when that happens, you get your whole sarcomere shortening. So here's a little video that's kind of cool showing you the calcium, and you can see all those little cross bridges forming, and they're pulling that actin and just swiveling like kind of like rowing a boat, or if you've ever done a tug of war. So see how it grabs and releases and grabs and releases and grabs and releases. So you have multiple cross bridge cycling that's going to cause that whole sarcomere to shorten. And when all the sarcomeres shorten, then the myofibrils shorten, and then your muscle fibers shorten, and whether it's a heart or whether it's skeletal muscle, the muscle contracts. So the overview of the whole process here is just that, again, you can see you have your neural first. So we have our action potential coming down. We have calcium coming in. So we need calcium in two places. We need calcium to release acetylcholine, and then we need calcium as well to remove the TT complex. We have to have acetylcholine. Acetylcholine is the excitatory neurotransmitter. Remember, that's what binds to depolarize the sarcomere and the sarcolemma. Then that depolarization comes down your T tubules, releases calcium from the SR. We talked about calcium moving the TT complex. Myosin grabs actin. The whole sarcomere shortens, and then you produce muscle tension. There's lots of videos that are really helpful. There's a BioFlix link in your helpful links folder. It's okay. It leaves out some details. If you want to start with that one, you can, but I would recommend you go into your mastering study area and go through all the interactive physiology. So the muscle contracts, but we don't always want our muscle to keep contracting. How do we stop it? Well, how does a muscle relax? Well, the nerve impulse stops. Remember, this all started in your brain. So your brain decides to stop it. And what happens is when it stops it, it causes an enzyme to be released, acetylcholine esterase. And so when acetylcholine esterase is released, it's actually going to break down acetylcholine. So that's going to come out and it's going to break down all the acetylcholine and get rid of it. Well, if you don't have any acetylcholine, and now these receptors are no longer activated. So it's going to stop the action potential. Well, if you don't have an action potential, right? Remember, that's what released calcium. There's no stimulus for calcium to be released. So now calcium is going to go back into the SR. And by the way, this requires ATP because you have to pump it. So you have to pump the calcium back in the SR. So we really need ATP for three things, right? We need it to cock the myosin clubheads so it can attach to actin. Remember, that's called forming a cross bridge. And then we need it to detach, right? We'll just say, let go of actin. And then we need it, thirdly, to pump calcium back in the SR for storage. So the calcium goes back in. It's just going to hang out there for the next time. Well, your key's gone, right? So now the TT complex is returned. Now we've covered up our active sites, so myosin can't grab it anymore. And so that's going to cause the muscle to relax because you can no longer contract. So we have this whole process of cycling and cross-bridge cycling, and those are the basic steps that are going to happen for muscle contraction. So I think it's really good to get in and watch some of those videos. Two quick things: muscle spasm and rigor mortis. So a muscle spasm, like a Charlie horse, this isn't a knot in a muscle. This is when the muscle, like large areas of the muscle, are actually contracting. So you're having like large muscle groups contracting. There's lots of causes, a lot of times dehydration, electrolyte deficiencies. So if you tend to get a lot of them, or calf spasms, make sure you get enough calcium, potassium, and magnesium. Those are the big three that you want to make sure you have enough in your diet. Make sure they have enough water. Muscle is about 75% water. Sometimes the nerves are too excited, or muscle fatigue can cause it, or lack of stretching. So those are some of the more common causes of muscle cramps. And when you get one, it's not fun, but you got to try to stretch the muscle so it can relax. And then you've probably heard of rigor or rigor mortis. So we're not memorizing this. I just wanted you to see that when you die, basically you have no more ATP, right? You're not producing ATP. And so what happens is the calcium doesn't get pumped back into the SR, and any cross bridges that are formed can't detach. So you have two things going on. You have some cross bridges still attached, and you're not getting rid of the calcium. So it stays in the cell. And when it stays in the cell, that's going to cause the muscles to contract. And they're going to stay contracted until the muscles actually break down. And that depends a lot on environmental conditions as they start to decompose. But rigor can sometimes be used to kind of give a sense of time of death. So this concludes our lecture on skeletal muscle physiology.